The overall goal of this protocol is to identify a monoclonal antibody-recognized linear B-cell epitope using serially truncated recombinant proteins and peptide scanning by dot-blot hybridization.
This method can help answer key questions concerning the molecular interaction between antigen and antibody. The main advantages of this technique are its simplicity and efficiency. The identified epitope can then be used in further investigations such as in therapeutic and diagnostic applications. Demonstrating the procedure will be Chien-Wen Chen, a postdoctor from my laboratory.
To begin, grow RGM56 mouse monoclonal hybridoma cells in hybrid serum-free medium. Maintain cell cultures in 175 T flasks at 37 degrees Celsius, 5% carbon dioxide. After 5 days of incubation, collect the culture medium after it is turned yellow and centrifuge the supernatant at 4500 times G for 30 minutes at 4 degrees Celsius. Then, discard the pelleted cell debris to obtain the antibody in solution. Next, add 2 milliliters of 50% protein G agarose slurry to a 5 milliliter column and equilibrate the resin with 10 milliliters of ice-cold PBS, equaling 10 volumes of preloaded resin. Load 200 milliliters of the previously prepared antibody solution onto the equilibrated column and discard the fraction that passes through the column. Using 10 milliliters of ice-cold PBS, wash the column twice. Finally, to elute the antibody from the resin, add 10 milliliters of